We are now starting part 2 of chapter 12 on the nervous system. Now, part 1 of chapter 12 for the nervous system ended on page 450. We are now starting on page 452 for part 2 of chapter 12 nervous system. It starts with this slide, slide number 51. So this is slide number 51. The title will be Plasma Membrane of Neurons. I don't care for the slide itself, so we move to the next slide. Don't care too much about this one either. I do care for this slide. Now, one of the things you learned in A&P 1 early on was that a cell membrane has channels in it. And these channels allow chemicals to go back and forth. Now we're going to find out that our mantra that we learned in the muscle chapter will return. And that mantra was electrical and chemical. So on this page, slide number 53, you'll see that there are two main channels. There's a chemically gated channel and there's a voltage gated channel. Now that's because we said that there's two ways muscles and nerves can be stimulated. Muscles and nerves can be stimulated either electrically or chemically. So muscles and nerves can be stimulated electrically or chemically. We also have a third one here that I'll talk about separately, and that is a leakage channel. And that leakage channel will be for potassium that we'll see later. Okay, next slide. This is a picture on page 452. This picture shows the different types of stages for the gates. Now these channels can be sodium channels, typically they are, or they could be potassium channels that we'll see also. Now remember in the muscle chapter we mentioned that there's these receptors, and these receptors act as channels, and when enough of the channel opens up, that a certain amount of sodium rushes in, and that certain amount was called threshold. Well, that same rule applies here. So a certain amount of sodium has to go through in order for it to stimulate and reach threshold. Now, we said that there's two ways. Electrically, we can stimulate these channels to open, or we can do it chemically. Right, next slide. Okay. Now this is the page I care for. This picture is found on page 454. On this page you see a sodium-potassium pump, you see a sodium channel, and you see a potassium channel. So these are the three main channels or pumps. Sodium-potassium pump, sodium leak channel, potassium leak channel. Now it does deal with something that I alluded to before in the muscle chapter. When we said that sodium rushed in, we call that depolarization. We'll see that momentarily. We also said that when potassium rushed out, we call that repolarization. And before, in the muscle chapter, we said that the combination of a depolarization and a repolarization is what produced an action potential. That is true here as well. So it's the combination of a depolarization followed by a repolarization that actually creates the action potential. We also saw in chapter 12, part 1, that the action potential only moves in one direction. It moves from the receptors all the way to the other end where the synaptic end bulb is. So it always goes in one direction. Okay, next slide. Don't care. Okay, this slide I do care, but it won't make sense yet. This is page 454 as well, but it's not going to make sense yet. Okay, don't care. Again, that won't make sense yet either. Now this is something we did already see. Matter of fact, we talked about this slide which is page 454 at the bottom, indirectly in the muscle chapter when we said that when the nerve action potential reaches the synaptic end bulb, that nerve action potential can cause calcium channels to open and allows for calcium influx. That is the exact same thing shown on this picture. So we have calcium channels there at the synaptic end bulb, allowing the calcium to come in only because of the nerve action potential causes things to happen. So in the muscle chapter, we wrote down 3 laws: refractory period, threshold, and all or none. We also said that a electrical action potential can move chemicals, and moving chemicals can create an action potential. That same thing with the muscle applies here as well. So moving chemicals can create an action potential, and an action potential can actually move chemicals as well. So that is, again, something we're going back to. Okay, next slide. Okay, now this slide's very important. Here we see it in words, but a picture form is on page 456, and you'll see that in a moment. Now what I'm getting at with this slide, to remember for the purpose of a test, is this thing down here called resting membrane potential. I don't care about memorizing the minus 70 millivolts there, but you do need to know that this is when the cell is at rest. So when a neuron is at rest, we call it the resting membrane potential. That's when the cell is at rest. Let's go look at the picture version of this. So the picture I care for is page 456, this picture here. Now the voltmeter, this machine here, is measuring minus 70 millivolts. That minus, that negative, tells me that the inside of the cell is negative. Now, before I go any further, there's some basics we have to get down. During a resting membrane potential, when the neuron is at rest, the outside of the cell tends to be positive. The reason why the outside of the cell is positive is because of the great amount of sodium which is outside the cell. So in this picture, page 456, you see a lot of these little red balls, and notice that the author labels the red ball as sodium, and you see a lot of the red ones only on the outside. That's because at rest the outside tends to be positive because of sodium. Now the inside tends to be very much negative at rest. That's why it says minus millivolts there, minus 70 millivolts, because the inside is so negative. Now the reason for why the inside is negative is because of the heavy amounts of protein. So if you look inside, you see a lot of big blobs here called proteins, and the protein makes the inside negative while at rest. Now you would say to me, Nimesh, I see a lot of these blue balls, and the blue balls are labeled as potassium. Now yes, despite the fact that potassium is a positive charge, and yes, the inside has a lot of potassium, regardless, because of so much protein on the inside tends to be negative, despite the fact that there's so much potassium is on the inside. So when you get a blood test done, they actually check for this kind of thing. They're looking for, where do I have more sodium, where do I have more potassium, and that's why the outside tends to be sodium positive, the inside tends to be negative, despite the fact that there's so much potassium inside. It's because of the proteins. Okay, so we have so much protein that it makes the inside negative. Now this is called resting membrane potential. Now if you can see here that we have sodium leaving, potassium leaving, and sodium coming in. We'll see about that in a little while. Okay. All right, so this is the typical way that we have the resting membrane potential. Okay, next slide. Now this is an important comment. This was briefly mentioned before in the muscle chapter. It's being brought out again. The role of the sodium-potassium pump. This is very important in maintaining the resting membrane potential. So after something happens in a neuron, how do I go back to rest? How do I get the dominoes to reset? Well, who resets the dominoes? The sodium-potassium pump. Now, the ratio mentioned here in the slide is important for a purpose of the test. Okay. It's going to be 3 sodiums for every 2 potassiums. So 3 sodiums for every 2 potassiums. Now, it's important to know the direction as well. So it's 3 sodiums out, and we'll see that in a little while. It's 3 sodiums out, 2 potassiums in. Now, did you ever learn this before in another chapter at the very, very beginning of the course, which was part of the general biology stuff? Yes, this was mentioned at that time. So it'll be 3 sodiums out for 2 potassiums back in. Okay, so one last time. 3 sodiums out, 2 potassiums back in, and the purpose of the sodium-potassium pump is to maintain the resting membrane potential. Very, very important. Next slide. Don't care, don't care, don't care, don't care. Okay, this picture, I do care. Now, take a moment and jot down this long term that you see here called excitatory postsynaptic potential, abbreviated as EPSP. Now, if we look at the picture carefully, we'll see that this neuron right here, having a synaptic end bulb, is releasing some neurotransmitters to stimulate the receptor on the second neuron. So this picture right here that I'm circling, that part is the first neuron. That's why it's labeled as presynaptic. This big blob over here that I'm circling is the second neuron, which is the postsynaptic neuron. So we have neuron number 1 stimulating neuron number 2. And it does it by releasing a neurotransmitter. So in each of these 3 cases, 1, 2, and 3, they're releasing some neurotransmitter that is exciting the second neuron. Now, how am I exciting the second neuron? I'm having enough of these neurotransmitters go in, and if you look carefully, you'll also see these red circles. These little red dots are your sodium, and it's mentioned in the illustration here. Sodium is going in, and if you recall, when sodium rushes in, we call that physically sodium influx, but electrically we call it depolarization. And that is what you see in this picture. We are depolarizing, stimulating the second neuron. And if you recall, if all of these sodiums go in, it's going to make the inside a little bit more positive. And that is why you see a bump there. That bump is letting us know the inside has become a little bit more positive because sodium has gone in. Remember, influx for sodium is the physical word. The electrical word is depolarization, and we're now making the inside a little bit more positive. And that's why you see a bump here. Now remember, the second neuron here is not going to fire unless I reach which is not done so far here, threshold. So if I just have a little bit of a blip like this here, this little bump, it's not going to be enough to reach threshold. So the second neuron here, the postsynaptic neuron, will not reach an action potential. And we'll talk about that as we go forward. So you have to have threshold in order to move forward. So we're basically stimulating the second neuron, but it's fizzling away because it doesn't reach threshold. Okay, that's EPSP. Next slide. Now we have the opposite here. This is now called IPSP. So jot down the full form. It's called inhibitory postsynaptic potential. So inhibitory postsynaptic potential, or IPSP, here what we're doing is we're doing the opposite. The first neuron, and I'll take this one as I did before, the first neuron still releases a neurotransmitter. But this time, the neurotransmitter does not stimulate the second neuron at all. Matter of fact, what it does is it allows two things to happen. It allows potassium to leave, and if you recall, potassium is what charge? Potassium is positive. So if positive potassium leaves the cell, it's going to make the inside of the cell more negative. So as potassium leaves, it's going to make the inside of the cell more negative. That means we're not likely to reach threshold, and I'll talk about that in just a second. The other thing that you see in the picture is chloride. Chloride is the Cl in the picture. Notice it has a negative charge. So if chloride goes into this second neuron, it's going to make that neuron even more negative inside. So as you can see from the graph, we actually have a dip now. We've made the inside of the cell far more negative. Now when you do that, it has a term that's called hyperpolarization. We're making the inside of the cell so negative that we hyperpolarize it. Now what's the importance of that? Well, if the cell is too negative, it's never going to reach threshold. And that means it's never going to get a chance to even make an action potential. You're actually making sure that that second neuron doesn't fire. And that's why this process is called inhibitory. So that's how certain things are actually stopped in our body. So certain nerves are stopped. So if you want to put your arm down and relax that muscle, we have to make sure that that motor neuron never fires. And the way we do that is hyperpolarization. In ANP2, how do you think the heart actually slows down? The heart actually slows down by hyperpolarization. We're making sure that the heart muscle doesn't get its stimulation, so it slows down. So hyperpolarization is making the inside so negative that it's not going to reach threshold, and the second neuron will not fire. Next slide. Okay. This is a combination. I don't care for this one. This one's confusing at times. Okay. Don't care for this slide. I do care for these two. Now these two are on slide number 78, and these two are ones that are important for us. I'll explain both of them one at a time. The first one is called spatial summation. In spatial summation, it's sort of like this. It's an issue of multiple, so you have many neurons stimulating a third one. But they're doing it differently. They're doing it in such a way that you have one neuron trying to stimulate a second neuron, a second one or a third one trying to stimulate one. So let's go back to that previous picture, and it's sort of like this example, where I have 1, 2, 3 trying to stimulate this one right here. Well, if I have 3 of them, and I get the first one to basically come close to making the second one here reach threshold, and then I have this one trying to do the same thing, and I have this one trying to do the same thing. If each one of them does it one after the other, you might have this big blob being our main second neuron actually fire. But that's called spatial, because it's 1, 2, 3, all 3 needing to work together in order to make this big blob, that our real second neuron, fire. Here's an everyday example. If you have one kid nagging at you, you may not respond to that kid. Then you have the second kid nagging at you right after it, or at the same time even. You might not respond to it. But if you have a first, second, and third kid nagging at you, you probably will have some response. That is your example for spatial summation, where multiple neurons need to work together to get the very last one to fire. So that's a spatial summation. Now let's go to the bottom one, temporal. Temporal is all about timing. So you have one neuron trying to stimulate the second neuron, but it does it one after the other. That means the first neuron tries to stimulate it, but the second neuron never fires. But instead of waiting between time, that's why temporal means time, it does it what? Rapid fire. So if Junior says to me, hey, I need my friend over, and then right after that, he says it again, I need my friend over. Third time around, fourth time around, fifth time around, but he doesn't wait. He doesn't let 10, 15 minutes go by. What he does is he fires one after the other, and eventually if you have that kid staring in your face, nagging at you 1 second after the next, after the next, after the next, you're probably going to take some kind of response. That's temporal summation, where it's all about the timing, the sequence of the timing, and that's what makes temporal summation effective. Okay, so temporal summation is all about time. Spatial is when it takes multiple neurons working together to make that happen. Next slide. Okay, here's a picture of what I've described to you. Here is a spatial summation. Let me give you a page number. This is page 462. So page 462. And it describes the same thing I did a moment ago. In spatial summation, you see many, many different neurons. In this example, there are 5 of them. And all 5 of them are nagging this big one right here, one after the other. So I have number 1, number 2, number 3, number 4. They're all nagging that big neuron, and it makes that big neuron eventually fire because it reaches what? Threshold. And that's shown by this graph here. The first one nagged, the second one nagged, the third one nagged, the fourth one nagged, the fifth one nagged, and that was enough to reach threshold. Okay, reaching that threshold. So that's spatial summation. Now comes temporal. This is one where I said that one kid is nagging the parent one after the other, after the other, after the other. And so notice this is one neuron nagging, 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 nagging until it reaches what? Threshold. And then that allows this big neuron to fire. So spatial summation and temporal summation. Okay, next slide. All right, now the slide we're on is slide number 81. This slide is something we've already talked about. That's all or none. The next one was threshold, and the third one was refractory period. So the 3 laws for muscles, the 3 laws for nerves are now mentioned here again. So I'll quickly say it before we move forward. So all or none, just for muscles and nerves, simply say that if a muscle is firing, it fires. If a nerve is firing, it fires. There is no halfway in between about it. So once I make an action potential, I either make an action potential or I don't. I either fire or I don't. A muscle either contracts or it doesn't. Okay? That's what all or none is. Threshold, we've already talked about. You got to reach threshold before an action potential happens. The other one was refractory period. So refractory period is what we mentioned before for muscles, where while a muscle is contracting, that same muscle fiber can't do anything further. Same is true for nerves. You can't have 2 trains on the same track. If a nerve is firing, you cannot make it fire while it's firing. And that's the threshold. Oh, sorry, excuse me, that's refractory period. Okay, next slide. Okay, now, this picture is very important to us. It is page 464 in sequence. Now, before I start this, there's something I mentioned in Chapter 12, Part 1. We said something about a place called axon hillock. And that was in Chapter 12, Part 1, talking about the parts of a neuron. So axon hillock. Now that is where a true nerve action potential starts. So a true nerve action potential starts at the axon hillock. That's very, very important for a test. Now why I mentioned that here is because this picture here is assuming that we're starting at the axon hillock. So this piece in red on the picture we're on, that little piece there is the axon hillock. Notice it says action potential there, and that's what we're starting with. Now, you can't see this yet, but I'll show it to you when I get to a different picture. But we have two kinds of potentials. We have an action potential, which is what I'm talking about now, and then in a moment I'm going to backtrack and talk about the graded potential. Okay, here we go. We're starting with action potential. Now in this picture we're starting with the resting membrane potential. Again, the page number I'm on is page 464. So here I have the the resting membrane potential, and notice the resting membrane potential is starting at -70, which is the resting state. In this situation, both gates are closed. That means the sodium gate is closed and the potassium gate is closed. Nothing is open because we're at rest. Notice where I see most of the sodium. Most of the sodium is on the outside, so the outside's positive, the inside is negative. Then, because of stimulation, we see what's happening. We see that the sodium channel, right here in the picture, where the cursor is bouncing, the sodium channel has opened. It's listed right there, opened. That allows some of the sodium to come in. So some of the sodium is now coming in. But notice, I still have not reached threshold yet. So I have to have some sodium come in, but it's not enough to reach threshold. Okay, next slide. On this picture, same page, on this picture, I see that I have what? I have allowed enough sodium to come in. Now the channel fully opens. And when the channel fully opens, I get a quick depolarization. Remember the language again. When sodium comes in, physically it's called sodium influx. That's when sodium physically comes in. But electrically we call it depolarization. So now sodium is rushing in, and it does it quickly. And it reaches a certain peak, as you see at the top here. Once it reaches this peak, the sodium channels will close, which is what we're going to see on the next slide. So now we see that the sodium channel is grayed out by the author. So the sodium channel has closed, because now it's grayed out, closed. What has to happen now is potassium will leave. So potassium will leave. Now as potassium leaves, we learned something already. As potassium leaves, the inside will return to negative. And as you can tell, as potassium is leaving, my number is becoming more and more negative. Remember, I'm going from positive to more and more negative. So now the inside is becoming very, very negative as potassium is leaving. Now the thing about potassium is that it stays open for a longer time. That's why it's sometimes called a leakage channel. So potassium will leak out. Now notice what happened. Because potassium stayed open, so much potassium left that we made the inside even more negative than it needs to be. It's even more negative than the resting membrane potential. That's a problem. We can't keep it that way. So in the next slide, we're going to see that eventually it comes back to rest here. Yes, it does say that the potassium channels close, but there's something else that the author has not mentioned here, but should be mentioned here. And that is this little rise back to rest was because of the sodium sodium-potassium pump. Remember mentioned earlier, who allows you to come back to resting membrane potential? The resting membrane potential is made possible because of the sodium-potassium pump, where 3 of the sodiums will leave and 2 of the potassiums will come back. And why is it called a sodium-potassium pump? Because it's an active transport pump where we're going against the concentration gradient. Okay, next slide. Now this is now page 465. It's a repeat of what we've done so far. Here is the flat line for the resting membrane potential. Here's where sodium is starting to come in by way of depolarization. Here is full depolarization because we reached threshold and the sodium channels open wide. Then we have the sodium channels closing at the top and the potassium channels opening, where so much potassium leaves that we overshoot the mark. And then that would be bad if we left it in this state. Because remember, that would be hyperpolarization, and we don't want to leave it there. Otherwise, nothing would ever happen. It's like stopping things. So what we got to do is bring it back to resting membrane potential, and that's done by the sodium-potassium pump. Very, very important. Not mentioned here, but it is something testable. So make sure you're aware that the reason why this line comes back to rest is because of the resting membrane potential. Okay, next slide. Don't care. Okay, I'm going to come back to this one in a moment. Now, so we'll come back to slide number 89, but there's something that needs to be mentioned, and I'm going to go back to our original starting point. Up here somewhere, which is where the dendrites are. So let me go to a better picture. Again, I'm picking a random picture, and that would be way up here where the dendrites are. And of course, the dendrites have receptors on them. Again, I'm picking a random picture. All I need is a neuron here. So here I have the dendrites with receptors. Notice where the axon hillock is. The axon hillock is right here where the cursor is bouncing up and down. That's where the action potential really starts, right here at the axon hillock. So Nemish, what happens up here? Well, all the way around here where the receptors are, that part is called the graded potential. So way out here will be a graded potential. The action potential only starts at the axon hillock. Now why is it important? Because a graded potential can be undone. A graded potential can be lessened. A graded potential can be enhanced. See, a graded potential has adjustability. So a graded potential can be undone, it can be lessened, it can be enhanced. But that's not true for an action potential. Once you form an action potential, it is always unstoppable. Assuming everything is normal, it's unstoppable. And an action potential always goes at whatever the signal says it's going to go at. So let's say we had something up here in the graded potential area that actually was numbing things. Let's say ice. You put ice on an injury and you're numbing it. Well, you're going to numb what? You're going to numb the graded potential. So you'll have less pain going down. And let's say it's at level 3. You know how doctors often say, what's your pain level? Let's say it's level 3 because of the ice. Well, that level 3 starts off here at the graded potential, and it only reaches this axon hillock at level 3. But once it becomes an action potential, it only stays level 3. Okay, let's go back to another example. Let's say here I have the graded potential that's at 9. Well then, at the axon hillock, it will be 9 all the way from that point forward. So the axon, this piece here, where the axon hillock starts, that's an action potential. It's fixed. It's unstoppable. Whereas a graded potential can be lessened, it can be enhanced, it can even be stopped. So if you actually don't have these receptors ever stimulated, then you get a 0. You get nothing. And then, of course, what do you send down when you have nothing? Nothing. Okay? So that's how a graded potential can be undone, it could be stopped, it could be enhanced, whereas an action potential is always fixed, unstoppable. These distinctions are important. Okay, let's go back to where we were. Okay, now I'm on this slide, and this one is called continuous conduction. Now what we're going to look at is this line right here. This is something I've actually already taught you, but I've never ever mentioned it until now. We've already said that unmyelinated neurons are what color from a previous chapter? Unmyelinated neurons are gray. Right? So unmyelinated neurons are gray. Well, they have what's called continuous conduction. And because they're unmyelinated and because it's continuous conduction, it's going to move very slowly. So continuous conduction will be found in unmyelinated neurons in the gray matter, and it's going to move slowly. So that's continuous conduction. The second type is called saltatory. Now the word saltatory gives the illusion that it means to jump. Okay? It really doesn't jump, but it gives the illusion that it's jumping from place to place, and we'll see that in a moment. So it gives the illusion that it's jumping. Now, saltatory is found in myelinated neurons. Now, we've already said in a previous chapter that myelination is a protein, right? Myelin's a protein. And the purpose of myelination before was to increase the speed of conduction and to protect the neuron. And remember, we had two kinds of myelinated neurons. Ones in the central nervous system were called oligodendrocytes, and the ones in the peripheral nervous system were called Schwann cells or neurolemocytes. So the neurolemocytes, the Schwann cells, the oligodendrocytes, they're all myelinated and they are saltatory. Now let's go see a picture on page 467 that refers to that. Alrighty. Let's see if I even have that picture. Okay, here I do. This is page 467, and we're going to use this one as our illustration. So page 467, we're going to see this middle part as our illustration. Now remember, this one's going to be for the saltatory conduction. Here we have our myelination as well. And notice, in between the myelination, we have something called node of Renvier. It was labeled in Chapter 12, Part 1, when we looked at the neuron. So when we looked at Chapter 12, Part 1 on the neuron, we saw the spaces in between the myelin sheathing called node of Renvier. Now, what is the value of this? Well, it increases the speed of conduction. That's why myelinateds are much faster. Now, how does it work? It works by having the action potential only trigger at the node. So this whole depolarization, repolarization, see how the author calls it nicely? Depolarization, repolarization. This action potential only occurs at the nodes. So when you have a myelinated neuron firing, it only flashes at the nodes. The illustration that I want to give you is a Mustang, a Ford Mustang. If you've ever seen the turn signal of a Ford Mustang, it's not like a typical turn signal where it just flashes. What a Ford Mustang turn signal does is it flashes from one bulb to the next in sequence, sort of like Christmas lights. We often call them chaser lights. So you have the first light bulb flash, followed by the next one, followed by the third one. So first, second, third, first, second, third, first, second, third. And that's your turn signal on the Ford Mustang. Same way here. We're only firing the action potential at the nodes. That is why some people call it saltatory, because it gives the illusion that we're actually stimulating by having the action potential jump from node to node to node. Sort of like what a stone would do if you skip it across a river or a pond. Right? When you're skipping rocks, it sort of jumps from one place to another. The same illusion applies here. We're giving the illusion that the action potential is going from node to node, making it much faster. Okay, next slide. Okay, I don't care. Don't care. Don't care. Okay, now this slide is something I mentioned already in advance. We talked about graded potential, action potential. This slide is the author's way of saying the same thing. So a graded potential can be undone. It could be enhanced. It could be minimized. But notice it says graded potentials only are local. So textbooks often call a graded potential the same thing as a local potential. So other authors would often call it a local potential or a graded potential. And notice it only travels a short distance. It can be undone. It can be enhanced. It can even not do anything. Whereas an action potential, the bottom paragraph, does always move forward. It's unstoppable. It moves at the same extreme. That means it doesn't weaken. It doesn't strengthen. It stays the same. So make sure you know the difference between action potential and a graded potential, sometimes called local potential. Okay, now the last slide that I have here, the one I'm on now, this slide talks about why we have a speed issue. Well, here's two things that make a conduction go faster. One is myelination, and the other is the thickness of the myelin. The thicker it is, the faster it goes. Okay? So the thicker it is, the faster it goes. Also, if it's myelinated, it does what? Goes faster. So what combination do you think would actually make the fastest possible? It has to be a thicker fiber. That means it's got to be bigger. And if it's myelinated. So a thicker one and a myelinated one would make it go much faster. Next slide. Now, the next few slides talk about the same concept, but they give you numbers. I don't want you to worry about the numbers. What I want you to understand is that you need to have both of them to make it the fastest. So if you have a thicker fiber or a thicker axon, and if it's myelinated, it's of course going to be the fastest. Okay, moving on. Okay. All right, now I'm on this slide, which is referring to page Okay, I'm on page 477. So that's where I'm at, 477. Now, for test purposes, I don't care about the slide outright. So I'm on page 477. Now here's what we need to do first. This one right here called acetylcholine, we saw that one in the muscle chapter, acetylcholine. But here's a catch. It may have been mentioned before, but it's now important to mention it to help you with A&P2. We said that acetylcholine stimulates skeletal muscles. So acetylcholine will stimulate skeletal muscles. But acetylcholine actually inhibits cardiac muscle. Okay, so let me say it one more time. Acetylcholine stimulates skeletal muscles, but it inhibits cardiac muscle. Very, very important. Okay, moving on. Okay, the next one I care for on this slide is GABA. GABA. Now, GABA is an interesting one. Okay, so GABA. Now, this is the one that's primarily inhibitory. So GABA is primarily inhibitory. So we have neurotransmitters that can be excitatory, and we have neurotransmitters that are inhibitory. Well, GABA is a key one that is inhibitory. So GABA being inhibitory. Okay, then I move up a little bit to glutamate. That one tends to be excitatory. Okay, so it tends to be excitatory. Okay, the next one I care for on this slide at the bottom here is substance P. This one deals with pain. So substance P, as in Paul, will deal with pain. So I've given you a couple. I've given you glutamate, which is excitatory. I've given you GABA, which is inhibitory. And then I've given you substance P that deals with pain. So those are the key ones that I will ask for, or more likely the department exam will too. Next slide. Don't care. Don't care. I've already mentioned this. Okay. I don't care. I've already mentioned this one as well. Okay. I don't care. Okay, this slide I do care. The term I care for is this one called facilitation. So this term called facilitation. It is something we've talked about but didn't give it a name. It's when neuron number 1 helps stimulate neuron number 2. That idea is called facilitation. So when one neuron has the ability to stimulate a second neuron, that's called facilitation. What do you think the opposite is? That is inhibition. So if the first neuron has the ability to inhibit the second neuron, that's going to be called inhibition. Very simple terms. So facilitation makes the second neuron more likely to fire. Inhibition ensures that the second neuron doesn't fire. Simple terms. Okay, don't care. Don't care. Don't care. Okay, now these I do care. We're going to do it by way of a picture on page 478. Here's the first one. So we're going to do one by one. The first one is called converging. Now these 4 that we're going to see in a moment are called types of circuits. So types of circuits. What I'm going to do is describe them, and that's what you need to know for the purpose of the test. So I'll describe them, and then that's what you need to know. The first one's called converging. Notice I have many, many inputs, but only one output. That's converging. That's all I care for. So many, many inputs, but only one output. That's converging. Next slide. Now this is the opposite. This is diverging. Again, page 478. The second one is diverging. I have one input, but many, many, many outputs. So one input, but many, many outputs. That's diverging. Okay, the next one is reverberating. Now this is a weird one. In this one, I see a whole lot of input, but notice the input becomes what also? An output leading back to the input. This is a loop. So this is like a feedback loop. The input partially becomes part of the output and then back to the input again. This is very important for learning, for memory. So reverberating circuits tend to be best for memory. So reverberating circuits for memory. Next slide. The last one is called parallel after discharge. So here I have one input, but as I have this one input, it branches off and becomes multiple inputs. So one input branches off and becomes multiple inputs. So you could look at it this way. One arrow splits into multiple arrows and then it joins back again for one output. So one arrow splits into many arrows and then merges back again into one arrow, and that would be parallel after discharge. Okay, all right, next slide. Don't care. And that's the end of chapter 12, part 2.